Did you know that the similarity between voltage-gated ion channel, ligand-gated ion channel, mechanically gated ion channel, and the thermally gated ion channel is that they alter the membrane potential once activated? They do so by changing their permeability for different ions in response to the presence of a stimulus they are made to detect. On the other hand, however, that instead of changing membrane permeability for ions, intracellular signaling molecules such as the cyclic adenosine monophosphate, inositol triphosphate, diacylglycerol, and calcium ions ended up being produced via the G protein coupled receptors, or that the bound receptor induces dimerization and protein phosphorylation. Sometimes the receptors may even be localized within the cell, in the cell cytoplasm or in the nucleus, so that bound receptor will initiate a change in the expression of a gene as a similar response. Let me first explain how does a G protein coupled receptor work. First, there needs to be a serpentine receptor with 7 membrane spanning regions weaving back and forth across the membrane. Next, there needs to be the G proteins, which are small membrane associated proteins consisting of the assembly of the alpha, beta, and gamma subunits. G alpha is normally bound to GDP, but once the serpentine receptor is bound, it changes shape and interacts with the G protein, causing the alpha subunit to release the GDP and associate with the GTP. The active G alpha subunit will then interact with either the cyclic adenosine monophosphate signaling pathway or the inositol triphosphate signaling pathway. For the cyclic adenosine monophosphate signaling pathway, once the alpha subunit of the GS protein binds GTP, it dissociates and activates adenylyl cyclase. Active adenylyl cyclase converts ATP into cyclic adenosine monophosphate. Cyclic adenosine monophosphate activates protein kinase A, which phosphorylates and modifies the function of a variety of intracellular proteins, namely enzymes, ion channels, and pumps. For the inositol triphosphate signaling pathway, once the alpha subunit of the GQ protein binds GTP, it dissociates and activates phospholipase C. Phospholipase C acts on the phosphatidyl inositol 4,5-bisphosphate to produce inositol triphosphate and diacylglycerol. While the diacylglycerol remains in the surface membrane, inositol triphosphate is released into the cytoplasm, binds to a calcium ion release channel in the endoplasmic reticulum, so that calcium ions floods out into the cytosol, binds to calcium binding proteins such as calmodulin, so that together they activate many enzymes and other intracellular effectors. Calcium ions also coordinates with diacylglycerol to activate protein kinase C, phosphorylating proteins involved in smooth muscle contraction and salivary secretion, for example. Once the GTP has been converted to GDP by the G-alpha protein's intrinsic GTPase activity, it redocks with the G-beta-gamma assembly in the surface membrane, awaiting for the next opportunity to bind to an occupied receptor.